dottoressa Magli sarebbe stata in grado di accendere il microfono. Si scusa per non poter essere qui e saluta tutti, ma ha avuto un imprevisto. Quindi moderiamo io e il professor Zuccotti. We have to switch to English because this is an international session.
It's a pleasure to start this new session. And the title is Future Avenues in Art Research. And the first speaker is Pasqualino Loi, who's going to talk about trends in veterinary research that will impact on human art. Thank you, Pasqualino.
Thank you, Maurizio. Good to see you again. And thank you, everybody, Andrea Borini, and for inviting me here, and the scientific committee for choosing me as a speaker. I guess you did this, both of you. The title, actually, I didn't choose the title. Giovanni Coticchio did, I guess. But I tried to arrange everything together to wrap up a nice story, and I will tell you what we will do. Actually, every time I come in these meetings, I realize that, I'm not doing anything important because, here people doing important job, making babies for other, couples. Whereas in my condition, just playing around, nothing serious, just enjoy ourselves. I think we are really privileged. In my next life, I will be a neurosurgeon. Should be okay, yes. I put here what I'm going to talk about so you can decide whether to leave or listen to what I'm going to tell you. First of all, I would like to update you with the current situation with using dry spermatozoa in our condition in farm animals. I'm a vet, of course, that's why I will be always working. What we hear now on is from farm animals, large animals. And next, I'm going to present something hoping to match your interest in something that we notice on our ICSI in sheep embryos that I think it might be useful for improving oocyte activation, even in your condition, in human embryos. And another one is the possibility to make artificial gametes from somatic cells. Actually, on my background, I'm an expert on embryo manipulation. I'm a cloner, so I'll be doing so all the time. And maybe there are some chances that we can actually channel this to the production of haploid gametes. And the last one is a little bit more daring. Well, I call it innocent mass trophoblastic exchange. Essentially, we're trying to make kind of universal recipients for holding, for carrying embryos from totally different genetic species. I call them precision chimeras, but let's see how you like it. As I said, I'm a vet. I've been working all the time with wild animals, starting with the mouflons and every kind of wild animals. And we are well aware of the situation taking place on the planet. We are facing with a very dramatic reduction on biodiversity, of course, owing to human-related activities. And dry storage is part of the enterprises just to make some biobanking for the animals we are going to lose. So, and then next we see what can you do with that genetic material. Of course, these banks were essentially, they were born and they still are like cryobanks. The largest one has been created by Michael Bruford, a very good friend of mine, by the Zoological Society in London. So I was gathering every kind of, genetic material from spiders, from fish, whatever actually is on the brink of extinction. And he got them stored in the frozen ark, he called it so, in London. But and then this paper was published in 1998. It was a kind of inspiring work for me. It really sparked a light in my brain. It was published by, the title in embryology. I'm sure most of you, the senior one at least, they know this scientist, okay? You too, I guess, Maurizio. Ryuzo Yanagimaki and Teruhiko Wakayama. Yanagimaki went to Japan. He squeezed out some spermatozoa from the epididymis of a mouse. They dried up in a normal lyophilizer. He put the vials with dry spermatozoa in the pocket. He took his flight back to Haiti, where they did this ICSI. and they got pups. That was, for me, was such incredible. I couldn't believe that. I started to do this using cells, but that's a long story. But indeed, actually, even that was a kind of obligate choice for me because it was impossible to get funded for my research on cloning. So that was another kind of line that was more, he paid actually in rewarding some funds. So with my group, actually, we steered toward drying of cells and, of course, gametes. As you can see here, if you compare the cryopreservation and lyophilization, lyophilization is much simpler. You can carry it out very easily, and of course, the most important fact is very cheap to keep stored. Of course, I'm talking to an audience of, expert cryobiologists. Nothing works better than cryo, so I'm not going to change. But I've shown this slide before, somewhere else. Maybe some of you know already this slide. But actually, I was very surprised because I made the discovery myself 2-3 years ago. This is Chris Polge. Chris Polge is the British scientist who's been the first one to freeze successfully full spermatozoa. In the very same paper, you can see here in the red circle, he said that he was frozen some sperm, but also freeze-dried another liquid of the sperm. So freeze-dry and freezing, they were born together from the very first paper in Nature, 1949, from the same author. So nothing new. So we started to do first with cells and then with spermatozoa. We really started to work intensively on these topics. The first surprise was to realize that, dry spermatozoa, I was expecting even drying cells. Now you add water, you rehydrate the cells, and then you think might be kind of soup. Instead, you could actually recognize clearly, if you see dried, rehydrated spermatozoa and fresh ones, you cannot say which one is the dry one. They really are perfectly okay. Of course, they are motionless. We need to do ICSI. We do ICSI using a piezo stepper device. We do it in a different way than you, which is much better, I think, in our condition. Because ICSI, as I'm going to tell you in a while, doesn't really work well. Yeah, so I started at the beginning working with Sefi Saragusti, a cryobiologist working with rhino and elephants in the Institute for Wildlife Research in Berlin. He was sending over across normal male this dry spermatozoa. And then actually I've anticipated because, 3-4 years ago, Wakayama did the same with mouse cell, dry cell, spermatozoa, selling them across by normal male. So that's another obvious advantage of having stored genetic material. Yeah, just to cut a very long story short, we actually are moving along. So freeze-dry was the kind of default methodology, but we found that you can actually dry cells with more convenient solution, technical solution. We teamed up in an European project with Imagen, okay, which is a company who does spin dry. Spin dry is a much milder procedure which removes the water by gently spinning under vacuum. And then they develop a wonderful system for packaging, really. It's a company, they took part in an European project, and they really bring a very big, important contribution to that project. Yes, as you can see, they have a special kit. You have a glass vial within a stainless steel one. You can actually label them with QR codes, so you can actually identify whatever you want. They see the size of the capsules. They are opening with a special device. And then you actually store them at room temperature. You really wanted to make the miracle, everything, and as soon as possible. You store them for 3, by even 4 years. I don't have the data in here, but trust me. And what was surprising to us that, if you see the expanded blastocyst rate, that's what you rate, the blastocysts, they were the same. Yes, 13% is the maximum number of blastocysts you can get doing ICSI in a sheep, in a pig, in a cattle, everywhere, but not in the horses. In the horses, everything works much better. But that's the situation in sheep. I'm going to tell you why in a while. So, of course, disappearing animals, they're not only the pandas or the tiger, but even ordinary sheep, small breeds, bred in particular situations are disappearing, That's a global problem. The FAO is actually looking for it, is trying to find solutions. So we store dry and cryo sperm from these two breeds. This is a special breed. I didn't know we had it in Italy, 4 horns. Yes, sometimes horns come in multiples of 2. That's a very, very unlucky sheep with 4 horns, but kind precious somehow. And there is a breeder which loves them and want to expand them as much as possible. So we are doing some embryo transfer and also some biobanking, okay? But if you look at reports that actually assessed how these dry spermatozoa can contribute to making an offspring, we have a lot of data in the mouse. A single horse was born and nothing else. So we decided 5 years ago to prove how far can we go, what is the potential of these dry spermatozoa to make into animals. That's the crucial aspect. So, yes, we did this study in Poland 5 years ago. We really wanted to make lambs. And to do that, we really didn't resort anymore to in vitro. You make routinely in vitro embryos in the sheep. But I really wanted to see what the dry procedure does on the offsprings. If you culture them in vitro, you have an additional horde of effects, epigenetic, stuff like that. I really have superovulated some sheep. I've taken the embryos, the oocytes, injected with dry spermatozoa, activated, and immediately back into the ovum duct. I really wanted to exclude all other possible sources of epigenetic variation, right? These are the first lamb from dry spermatozoa in the world. The dry spermatozoa were dried at minus 80 degrees, okay, stored for 2 months, and they were born, over Christmas. We need a lamb for Christmas, right? First 3, 2 girls and a boy. Second group, again 3, 2 girls and a boy, born 2024, this time stored dry at minus 20. We had another large group in the summertime. It is the first scanning we did in September. And then this dry sperm has been stored at 4 degrees, as you can see on the slide. Okay, so that's very important to store them at plus, no minus. We didn't publish this because I want to see how these animals are growing. What are the behavior? I have an expert co-worker of mine which is able to see whether a sheep is autistic or schizophrenic. I don't know whether to trust him or not. But yes, they are doing a lot of metabolic follow-up, clinical. The vets are taking care of these lambs, just to see. You are running glucose tolerance test just to see whether a kind of diabetes, body weight. There is a specialist, taking care of all the cardiophysiology of these animals. They are normal under any parameters. But you really wanted to go even further, right? And we made also second generation sheep. So the first which are born have been bred with normal other animals. We are now the second generation. And again, you're actually checking for any kind of behavioral, metabolic, or kind of clinical problem. Okay, I really want to be sure that these animals are normal under every point of view. So we got a couple of European projects on these topics. I'm very grateful to all my partners and to my people because we really advanced a lot, as you could see, the state of art of dry gamete preservation. Whether we are interested or not, I don't know. We launched a company, a startup with my partner, my group in Teramo, and then we got founded for European project. It's called InnoWide, InnoDry, to see whether Japanese clinics are interested in dry spermatozoa storage. They say no. So I don't know whether it might be useful. In our case, for biodiversity preservation, indeed, it is important. So that's the second point I wanted to touch with you. As I told you, ICSI in large animals doesn't work. And then, so the situation is the one you can see. 85% of them are activated because they have two pronuclei. They never migrate each other. They never undergo syngamy. So I've asked Luca Palazzese, try to understand where the problem is, and then he just always found the same. You have, on the upper row, embryos fertilized, zygote after fertilization, and after ICSI with spermatozoa. Those down you can see pronuclei never met, never fuse each other, so that's the major problem. And then I say, why? He did a lot of study with another girl, which works with me again, Luisa Gioia is the name, and she did, this calcium pattern monitoring in ICSI and normal oocyte. There are no such studies in the sheep, unfortunately. And then he noticed that in the sheep, you have a normal, for 24 hours, cyclic release of calcium. When you do ICSI, you do ionomycin for 5 minutes, and then 3-hour cycloheximide is a protein synthase inhibitor. You have just one single spike, okay? Which you don't have the other spike of calcium, which actually activate the cytoskeleton and then induce syngamy. That's the problem, okay? Why I see that may be useful. Lucas provided the answer, has published the answer to the question a couple of months ago on Pterogenology. Why is important? Because I think if we give these zygotes, calcium every hour, I'm expecting that everything will be actually be developed better. But I need somebody if you're working on, I guess, what is called microfluidic to address the problem. Okay, so artificial gametes for direct haploidization of somatic nuclei. 5 minuti, oh caspita. Yes, a part of this, this animal, there are no one living. There are 3 white rhinos. They are all disappeared. They died last year. And we organized a kind of rhino group with Cesare Galli, Thomas Hildebrand is the guy here. Okay, and Ian is the vet who does the pickup in the rhino. We should have decided to approach a rhino, trust me. So we started to see, it's a matter of bringing them back to life. And so the option is to make even artificial gametes is possible. And luckily, Cesare Galli in particular and Giovanni, they have established the first embryonic cell lines in the rhino. And you have this guy, Hayashi, is a top scientist, which is actually been the first one to make oocytes and spermatozoa from sperm using, embryonic stem cells from the mouse. And then you have, reporter genes. That's the kind of scheme. There's light up when you have a stella horn, which is the first gene for primordial gersema isolation. He picks them up, he puts them together, but you cannot do that in a rhino. Even in human, you can never be able to do that. That's okay for as a model, but it's impossible to make any kind of clinical application, or zoological application, if you like. This paper was published this summer. Actually, I've reviewed this paper for Nature Communication, and actually I had quite a large echo because there was a commentary on Corriere della Sera by Carlo Alberto Redi, your boss, Maurizio. And then they say, no, yes, we have human embryos from, haploidization. Actually, that was over-optimistic, I guess, because, yes, they were able to inject the cells, and then the haploidization was quite perfect. 23 chromosomes in the polar body, 23 chromosomes reiterating in the oocytes, and they used this clamp to fertilize another egg. Okay, but I said these are somatic chromosomes. They will be never able to make a lamb. It's a kind of semi-cloning. You need to have a haploid set of chromosomes. They must be totipotent. Otherwise, there is no story. You see, that's a cloned embryos. I know these embryos very well, ladies and gentlemen. These embryos is going nowhere. It will never develop. So, and then you need to do something to make them totipotent. This paper was published again a couple of months ago. I'm very happy because it has kind of validated something that I've done with my group. They did artificial gametes. Okay, look, they have removed the chromosome from the oocytes. They made androgenetic embryos, injecting a single spermatozoa, and then you have haploid stem cells. Animal, sorry. They did in bovine and in the sheep. And they have haploid, embryonic, haploid, yes, androgenetic stem cells, okay? Then they have these cells, they did genetic modification by CRISPR-Cas9, and they use these stem cells haploid to fertilize another oocytes, okay? They got lumps only if they use protamine cells. So they have taken these haploid stem cells, they induced in them the expression of protamine. The genome is protaminized, as you can see in this slide in here, and then only in this case you can have fully totipotency and you have, the offsprings. So I'm very happy. I don't want to, because you see on the top line, don't watch this very, very messy slide. Don't watch everything. You see, but at the bottom line, you see the expression of protamine actually compacts the nuclei just like a spermatid cell. This actually displaces away all the 3-methylation of histone 3, lysine 9, lysine 27, lysine 4, actually cleans. I don't want to go because, I don't want to be late. I'm already late. This work has been done by my group, so I'm very happy that actually somebody else was able to validate till the end. So you look at, these are normal cells, protaminized ones. They really look like spermatid. And that's the only way to induce a kind of totipotency. So no now, but if you protaminize them, yes, maybe you'll be able to have totipotency. So this, I will just close. I'm sorry, I'm running a little bit late, but I'll be very fast. You want to make the sheep, in this very old paper, a sheep gave birth to a goat, which is impossible. The embryo resulted from an aggregation from blastoma from a goat and from a sheep. For somehow, God knows what happened, so you have an innocent mass from the goat, and you have a kid, and then you have the placenta from the sheep. We want to make them in the horse, because, even if you are able to make a lot of horses, embryos, where they want to transfer. So are you able to do, to dissect, as you can see here, we have a horse embryo, you remove the innocent mass, you leave the trophoblastic vesicle, you can see in that line there. You do the same in the horse, you keep in the rhino, so you take the rhino innocent mass, you transfer that into the trophoblastic vesicle, and then you have an hybrid embryos you transfer into the sheep, into the horse, and the horse will deliver a rhino, okay? That's long-term kind of science fiction. So he started to do it in the sheep. Okay, so you want to transfer precision chimera where you have the trophoblast made from the sheep. These are raw DNA embryos. You can see you cut off the innocent mass. That's very, very quick, very fast. You inject the innocent mass. That's more tricky. That's really a kind of invasive manipulation. And then you should have a precision chimera where the sheep can carry the pregnancy because, to traffic to them interacts with the uterus, and the sheep will deliver raw deers. That's going to be my last experiment, I guess. I want to conclude. I apologize for being a little bit late. Yes, I think dry storage in spermatozoa works fine. Don't know whether it might be useful for human reproduction. We have a project with Donatella Paoli with this. And I'm very, very glad. Actually, I thank her very much because she's pushing forward the field and doing some precious work on dry human spermatozoa. I like very much to do some sheep work on the activation. I want to repristinate a kind of calcium wave. I'm sure it will normalize the embryo. Artificial gametes, I think, are not really, that's not the time. And then I'm going to conclude. Sorry, I don't like to go out over my time. I apologize again with the chairs. Yeah, that's, the people that work with me, and these are the guys that give us some money to do these experiments. And again, one other print, I'm coordinated by Donatella, which is doing the same work on the human spermatozoa. Thank you very much, everybody. Thank you.
Thank you very much. So, Lino, we are going to have a discussion time at the end?
Sir.
Okay, thanks.
So it's my pleasure to introduce the next speaker. It's going to be Maria José de los Santos. No need for introduction. She's going to talk about autologous mitochondrial transfer. Thank you.
Thank you for the introduction. Hello, everyone. Good afternoon. It's a pleasure to be here today in this symposium. I would like to thank the organizers for counting on me to present this data about autologous mitochondrial transfer, what's new. I have to say that there is not that much, so I'm sorry for the spoiler, but I think it's a good opportunity to review what has been done in this topic and what we are right now. It's true that the benefits of addition of extra number of copies of mitochondrial DNA into the oocytes during the injection or using other assisted reproductive technologies has been always a matter of controversy, not only because there are some, let's say, ethical concerns from some members of the society, but also because there are real doubts about the efficacy and safety. There are many things that we need to understand what happened when we do a mitochondrial donation in terms of the balance between the mitochondrial genome and the nuclear genome. Is there any effect in terms of epigenetic level in the offsprings? So what I'm going to do today to discuss about this topic is to make a brief review of about general aspects of mitochondrial replication in the embryo and oocytes. I would like to talk a little bit about the concept of supplementation of mitochondria to restore oocyte competence and what are the mechanisms behind this explanation, possible sources of autologous mitochondria. I would like to share with you a little bit of the experience doing augmented mitochondrial transfer with autologous from egg precursor cells, autologous mitochondria from egg precursor cells. What we know about the children follow-up is very important to know the health of the babies that has been born from these procedures, and I will conclude with some final remarks. So mitochondrial and mitochondrial copy number are very important in embryo development, and perhaps this is why the number of mitochondria in the oocytes are so abundant. It's the most abundant organelles in the oocytes. But the number of mitochondria in the cells is not always the same. It depends on very many things. It depends on the cell type. It depends also of the cleavage stage of the embryo, and also it depends on the metabolic requirements from the embryo. So let's start by the very beginning. So from the germinal cell line to the final maturation stage, there is like an increase of mitochondrial copy number from the primordial germ cells to the final maturation stage. So we start with a very few number of copies in the germline, the primordial cells, and this few number of mitochondrial DNA would serve as a template that is going to be transmitted to the germline. But even though at the end of the maturation, we reach the peak of the maturation of the mitochondrial DNA copy numbers, it's true that when you compare the mitochondrial mass between oocytes from the same patients, we can find important differences. So we might find difference between oocytes from within the same patient, and of course there are big difference between the mitochondrial mass of oocytes coming from different patients. This is one thing. And the other thing is that if we look at the oocytes that fail fertilize, in the majority of the cases, we are going to see a low mitochondrial copy number. Now, what happens if the fertilization goes well? Well, from fertilization to blastocyst stage, what is going to happen is that there is no replication whatsoever, or maybe a minimal replication during this stage. So that means that if we make a calculation of the mitochondrial copy number per cell in the embryo, the number of mitochondrial DNA per cell will be reduced as the embryo divides, although the total mitochondrial mass in the embryo is going to stay the same. Now, if we want to make a more detailed observation of what happened with the mitochondrial configuration in the blastocyst, we find very interesting things. One thing is that at the very beginning, at the blastocyst stage, we might find that the mitochondrial copy number in the inner cell mass is much higher than the introfectodermal cells. But what happened is that all of a sudden, the mitochondrial, as the cells of the inner cell mass start to multiply, to divide, the mitochondrial DNA will be diluted out in the inner cell mass, and the DNA replication, the mitochondrial DNA replication, is going to be confined to the trophectoderm cells. The difference is not only about the number of mitochondria in the inner cell mass and the trophectoderm cells. We also know that if we look at the configuration of these mitochondria are different if in the trophectoderm cells compared to the inner cell mass. What are these differences? These differences are related with the activity of the mitochondria. If you see this part of the graph, let's say here, you can see that the distribution of the mitochondria in the trophectoderm cells are different from the distribution of the mitochondria in the inner cell mass. And as you can see, this difference in distribution is because there are much higher number of high activity mitochondrial and moderate mitochondria in the trophectoderm cells. Now, why mitochondria are so important in cells, in embryo development? Well, because these mitochondria, these organelles, is like integrates a huge number of processes that are very important for cellular functioning. They are not only important for ATP production through the tricarboxylic acid cycle or beta oxidation of the fatty acids or catabolism of the amino acids, but it's important that many of these metabolites also have an effect on the epigenetic level. So those components may also make changes at epigenetic modification. We can increase DNA methylation, and also thanks to these metabolites, we can also make some changes in chromatin and the protein and chromatin. We all know the importance of the mitochondria in cell signaling, in apoptosis, in the creation of reactive oxygen species, and all together is make these cells to function. But what happens in the oocytes that are considered bad quality? Well, when you look at the oocytes that are considered bad quality, what happens is that there are a huge amount of abnormalities in the mitochondria. What kind of abnormalities I'm talking about? I'm talking about a decrease of the mitochondrial mass in oocytes considered bad quality. Also, many times we can see a disruption in the membranes of the mitochondrial, low ATP production. Sometimes we have seen a lot of mutation and deletions of the mitochondrial DNA, and all together may affect the mitochondrial dysfunction that will affect, of course, important aspects during fertilization and embryo development. So we are going, if the mitochondrial does not work properly, we are going to have problems during the exocytosis of the cortical granules, or during the membrane formation, during spindle formation, chromosome segregation, and of course, embryo development. Now, having this in mind, what can we do to fix oocytes that are bad quality? Well, one of the strategies that were explored was the implementation of the donation of autologous mitochondrial for other sibling oocytes. So back in 2016, there were some studies in pigs where demonstrated that the implementation of the donation of good quality mitochondria into oocytes that were not considered good quality were able to improve embryo development. Thank you to this thanks to this supplementation, they were able to demonstrate that in all the stages, the mitochondrial DNA copy number was higher, and also that blastocysts derived from these treated oocytes have the same gene expression patterns that the control oocytes, which of course were very interesting. Now, what is the action that, what is the rationale behind the DNA supplementation? Why DNA supplementation is going to help to restore oocyte quality? Well, it seems that it depends on a feedback loop between the mitochondrial DNA copy number and the promoter of Pol gamma. How it works? When we have a bad quality oocyte, that means that we are going to have low number of mitochondrial DNA, either because of stress or because of damage, this low numbers of mitochondrial DNA is going to send signals to the nucleus, and then all this cascade of the methylation of the promoter of the Pol gamma will make the Pol gamma to work, to transcript, and restore the mitochondrial DNA copy number. So if we make the DNA supplementation with good quality mitochondrial during this narrow moment of injection, we might take advantage of this feedback, this loop, to restore the mitochondrial, the bad quality mitochondrial by good quality ones. Now, probably you are aware of all these attempts trying to improve oocyte quality and pregnancy rates to alleviate infertility using different strategies. And probably you also know that when these strategies, ooplasmic transfer, spindle transfer, were used, we were observed some interesting situations like heteroplasma and also some maternal reversal. So what can we do to, let's say, control this, or to control this drift of mitochondrial? Well, the only way is to use autologous mitochondrial transfer, mitochondrial sources. So some of the experiments of some of the data that we have using autologous sources in the animal model demonstrated that the cells with higher performance were, let's say, stem cells. So as you can see in the table, the use of adult hepatocytes or even the platelets were not able to improve neither fertilization rates, neither blastocyst rates. However, the use of stem cells in the mice, either young or aged, were able to increase the blastulation rate and the lipid rates. So maybe stem cells have something to do. What happened with the same? Do we have some experience in the adult, in the humans? Of course we do, and there are some studies trying to use granular cells to increase the blastulation rates and pregnancy rates. And as you can see here, some studies using cell granulosa cells, mitochondrial from cell granulosa cells, were able to improve the blastocyst formation rate and also the pregnancy rates. Also, we have some experience with using bone marrow mesenchymal stem cells, and the same, they were able to see some improved in the embryo quality and, of course, a little bit of increase of ongoing pregnancy rates. Other cells that has been used, well, the so-called ERB precursor cells. What are these cells? Probably there are so many different opinions with regards of the existence of these cells in the ovarian cortex, but the point is that when they were used for mitochondrial transplantation, there were some studies claiming that this transplantation, the use of these mitochondrial from these stem cells were good enough to improve pregnancy rates. So far, the studies that has been used these egg precursor cells are these ones. The first one, I have to say that in the majority of these studies, they were not very good design because in most of the cases, the results were compared with previous outcomes in the same patients with previous outcomes. But the truth is that using these strategies, people were claiming to have very good, well, at least much better pregnancy rates than when patients did not use any other strategy. And we also participate in a study because we thought that these studies were not very well done, and we wanted to really test whether the use of this mitochondrial, good mitochondrial from egg precursor cells were able to improve clinical outcomes. So what we do is to make a randomized control trial patients with different IVF failures, and we randomized the oocytes from the sibling oocytes. Were randomized, and we treat oocytes with this mitochondrial supplementation versus the control. And as you can see here, we were not able to see any difference at all in terms of fertilization or embryo euploidy. Actually, when we compare the blastulation rate between the control and the treated group, we have better blastulation rates in the control groups, but better embryo quality in the oocytes that were not treated. Again, we didn't see any difference in all the morphokinetics aspects of the embryo, and perhaps some small difference in terms of the pronuclear appearance, maybe because of some boost of the mitochondrial DNA during the first stages. And when we look at the mitochondrial copy number in the trophoctodome cells of these blastocysts, even though we have some increase of the mitochondrial copy number in the trophoctodome cells from the treated group, the difference were not significant. Here there is some results regarding the comparison between the control and the augment, and as you can see, we were not seeing, any improvement of the pregnancy rates where blastocysts derived from augment group were used for transfer. Oops. Okay. So what's new? I think it's a good practice to know about the safety of the babies that has been born with these practices. And we did a follow-up of these 18 children, and as you can see here, we were not able to see any difference in terms of obstetrics variables or some neonatal outcomes of the embryos of babies that were born from both groups. Likewise, some studies made from the Japanese group that were also using augment treatment were not able to see any congenital abnormalities on the babies born from these strategies. And when they were doing some, let's say, motor and cognitive score, these babies were totally normal. This Japanese group were trying to do the correlation between the mitochondrial copy number of the babies and the concordance between the babies and the mothers. And in most of the cases, in a small number of patients, were able to see a huge concordance, except with this baby that has a 99 concordance with the mother. And they claim that is within the normal range. Now, as I conclude, mitochondrial transfer was proposed as an innovative third therapy to restore oocyte competence. The use of autologous was considered a good option to avoid heteroplasmia. But from a list of different cellular sources of autologous mitochondrial, stem cells seem to be the best candidates, because those mitochondrial cells coming from these stem cells are more immature, they are more quiet, and it seems that they should be able to restore bad quality mitochondria from bad quality oocytes. It's true that some of the candidates that were explored in the human side was this called egg precursor cells, and we don't know whether they exist or not for sure. So far the live births has been achieved using this autologous mitochondrial transfer from non-randomized control trial. They were okay, but the randomized control trial demonstrated that the augment treatment did not improve the live births of the patients. From the safety point of view, I would like to point out that also the augment treatment was not very safe, because it was this technique required a laparoscopy to take some pieces of the ovarian cortex, and it was not very good for the patients under my point of view. And for the short-term follow-up of the children, we have to say that so far there is any indication that the babies have some imprinting defects of other congenital abnormalities compared with the control groups. Other approaches using autologous mitochondrial are limited, are not yet considered feasible, and we don't think that with the current knowledge we can say that this strategy can be used for alleviating fertility in our IVF patients. So far, I think is all I wanted to say. Thank you very much for listening. And, well, I don't know if there are any questions.
Thank you. Thank you very much for sharing your data and for a very interesting talk. Thank you.
Thank you, Maria José. And we move on to the next speaker, Sonia Herraiz. Hi, Sonia. And her talk is titled Hopes and Challenges of Ovarian Reactivation.
Well, good afternoon. First of all, I want to thank the scientific committee for inviting me to this amazing symposium. We will just review a little bit this ovarian reactivation field. Okay. The last century was characterized by the population growth, but this one is meant to be for the population aging. Indeed, it's been estimated that by 2030, 24% of European population would be over 65 years old. And this is especially relevant for women, as most of the human organs start to show the first clinical signs of aging when we are around 50, 60 years of age. But the ovary, as you can see in the graph, shows a unique decline in reproductive function starting when women are 35 years old. Ovarian aging, this is really well known, associates decreased ovarian reserve, but also impaired oocyte quality by increasing aneuploidy rates, prevalence of genetic abnormalities in the newborns, and infertility from 38 years of age onwards. However, I want to highlight that even when the ovary loses the ability to ovulate due to menopause, or in the worst ovarian situation of premature ovarian insufficiency due to a cancer treatment, for example, in the ovary remains a residual pool of dormant follicles that can be reactivated to growth. And indeed, this residual pool of follicles has been the base of all the ovarian reactivation techniques that have been developed during the last 10 years. The first one is the ovarian fragmentation, which is based on the original in vitro activation technique of Kawamura, that by using the mechanical fragmentation of the ovarian cortex inhibits the Hippo pathway that at the very end promotes follicular growth. Different results have been achieved by this technique, but the unique randomized clinical trial testing the ovarian fragmentation in poor responders showed that the technique is able to improve the development of the follicles, but when the IVF outcomes were compared with controls, they do not really modify. But in this same paper, they shown that for a specific population of young women with intermittent premature ovarian insufficiency, the ovarian fragmentation leads to a 41.6% pregnancy rate, which is a really good result for this population that has no other option than egg donation. The second technique, which is one of the most widely used, is the intraovarian injection of PRP. This one raises a lot of controversy because the results are very heterogeneous. For example, in poor responders, we can find pregnancy rates ranging from 20% to 46%, or 10% in POI population. And finally, I just want to mention the use of stem cells that finally led to the ASCO technique from autologous stem cell ovarian transplant developed by our group that uses bone marrow stem cells to reactivate the ovaries. And in the first pilot study that we developed in poor responders, we found that the use of this technique allowed a 33% pregnancy rate in a long-term infertility population that was not able to reach any single pregnancy in their previous attempt. However, what I want to highlight is that all these techniques focus on promoting follicular growth, and they retrieve different efficacies depending on the patient diagnosis. But none of them modify embryo quality, because all the studies, including PGTA, showed aneuploidy rates according to the patient age. And this means that the oocyte quality issue still remains a challenge for us. But to develop any treatment or screening tool, we first need to really understand the ovarian pathologies, their causes, and the underlying mechanism. And with this purpose, we develop a study where we collect peripheral blood plasma from fertile patients and from patients with POI. Using high throughput protein techniques, we were able to identify and to quantify all the proteins present in plasma in order to check if there's any difference depending on the ovarian phenotype of the patients. Interestingly, what we found after assessing the plasma proteomes is that there were 124 differentially expressed proteins between fertile patients and POI patients of the same age. And this is very important because it is known that as we age, the composition of our blood changes. When we are young, we have plenty of growth factors, and as we get older, all the pro-inflammatory proteins start to raise. So as they are the similar age, these differences could be due to the ovarian phenotype. Indeed, the functional enrichment analysis showed that in POI, the complement and coagulation cascades were upregulated, while oocyte meiosis and the activation pathways PI3, K, AKT, and HIPPO signaling pathways, among others, were downregulated in POI women. 60 of these differentially expressed proteins are involved in what has been considered as the hallmarks of aging, of the ovarian aging in this case. And just let me highlight, for example, cellular senescence, genomic instability that at the very end will lead to aneuploidy, or aggravated inflammatory response. This data provide us with really nice information that would allow us to identify new pathways to improve oocyte quality and aneuploidy. But how can we move from this situation to this one? Well, it is well known that the oocyte quality and bioenergetics, which are key factors for future embryo development, are decreased with age, but also with the disease. Indeed, DNA damage response and repair, metabolism, and apoptotic regulating pathways are key factors controlling ovarian aging. Recent studies show that the double-strand breaks, which are the most deleterious form of DNA damage to the oocyte, leading to aneuploidy, can also be generated by standard physiologic or metabolic stress. Indeed, single-cell RNA-seq experiments show that oxidative damage is one of the main threats for the ovarian cells, because impaired mitochondrial bioenergetic increases the reactive oxygen species levels, apoptosis, DNA damage, and spindle instabilities during chromosome segregation that will affect oocyte quality and embryo euploidy. But how can we efficiently improve the oocyte bioenergetic system, which seems to be so important for final embryo development and euploidy? Well, recent studies suggest that nicotinamide mononucleotide, or NMN, supplementation reverses the decline in female fertility associated with aging. NMN is a precursor of NAD, which is an important cofactor for all living cells involved in essential biological processes such as redox reaction or DNA repair. In the context of ovarian aging, in 2020, the group led by Bertoldo published a paper where they describe that the NAD decline during reproductive aging is associated with oocyte dysfunction, meiotic defects, and impaired embryo quality. And in this paper, they test how the oral supplementation with NMN, this NAD precursor, was able to restore oocyte quality in aged mouse and improve embryo development and the reproductive outcomes. But these are mouse experiments. Physiological aging in the mouse do not always mimic what happens in women, because mouse do not develop aneuploidy. And aging is not the only cause of fertility decline. Based on that, we start a new experiment using a more deleterious mouse model of ovarian damage. In this case, we designed 2 animal models mimicking diminished ovarian reserve and POI conditions by using cyclophosphamide and busulfan, which are 2 of the most damaging chemotherapeutic drugs for the ovaries and the oocytes. Once the ovarian damage was established, we randomized the animals to receive oral supplementation with this NMN molecule or to control during 4 weeks. This time frame means in the mouse that we will cover a whole folliculogenesis cycle. So we will see the effects, if there's any, on the primordial follicles exposed to the damage. After the treatment, animals underwent ovarian stimulation, and we analyzed all the effects on the ovary, but also on the metaphase 2 recovery. The results of these experiments were published early this year in the American Journal of Obstetrics and Gynecology, and the first thing that we found was that there was no effect on the oocyte number or on the follicular number, in contrast to what Bertoldo found using this physiological aging model. But interestingly, NMN supplementation was able to reduce all the morphological abnormalities in follicles and metaphase 2 eggs that were induced by chemotherapy. Indeed, the first thing that we did was to really check that the oral supplementation that the animals were receiving was able to reach the oocytes. So we take advantage of the autofluorescence properties of the NAD molecule, and by using FRET microscopy, we were able to measure the NAD levels in live metaphase 2 oocytes. And as you can see here in the upper panel, chemotherapy really impairs NAD levels in the oocytes. But by providing our animal models with this NMN supplementation, especially in the diminished ovarian reserve and the POI animals, when compared to their own control, the NAD levels were improved. NAD is highly related with reactive oxygen species, so we then check how the levels were. And as you can observe, again, chemotherapy is damaging a lot the oocytes. You can see how bright the POI eggs were. This means they have a lot of reactive oxygen species. And in this case, especially in this more damaging condition, we observed that NMN oral supplementation for a whole folliculogenesis cycle was able to reduce the ROS levels in the metaphase 2 oocytes. But, I mean, this is fine, but we are really interested in euploidy. And the meiotic spindle plays a key role to separate the chromosomes. So we first analyzed how the conformation was, and as you can see in the upper panel, compared to controls, the chemotherapy treatment completely destroys the spindle. But after providing this NMN supplementation, even in the worst ovarian situation of POI, the eggs were able to have a normal spindle. Indeed, when we measure all these parameters to have really quantitative data on that, we found that NMN enhanced spindle assembly and kinetochore microtubule junctions in both animal models. And another interesting result that we found was that in young healthy animals, NMN supplementation seems to induce some deleterious effects on the spindle formation. Then we moved into how the eggs work, and we performed in vitro fertilization of some of them. And as you can see here in the table, NMN was able to improve both the fertilization and the blastocyst formation rates in both ovarian damage models. And again, we saw some deleterious effects of additional supplementation in young healthy animals. But these are really nice results, but we are working with mouse, which makes that everything is shorter. But a human primordial follicle requires up to 5 months to reach the antral or preovulatory stage. And sometimes we do not have such an extended period of time to initiate the assisted reproductive techniques in our patients. So to try to explore more on this possibility, we perform a second experiment using exactly the same animal models and the same treatment, we reduce or we limit the NMN supplementation to only 14 days, which means to target the later stages of folliculogenesis. In this case, as we observed in the first experiment, NMN was able to increase NAD levels, to reduce ROS abundance, and also to modify or repair the mitochondrial distribution that was seriously impaired, as you can see here, by chemotherapy treatment. And after NMN, even in the POI eggs that were, run out of mitochondria, we were able to have a similar pattern to the young healthy animals. These changes came across with promotion of the DNA repair genes in both animal models, as well as improved fertilization and hatching rates in chemotherapy-damaged oocytes, suggesting that NMN may have restorative effects on oocyte quality after chemotherapy-induced damage, offering new potential options. This is amazing, but again, this is an animal model, and we need to validate these results with human eggs. In this case, we made a slight modification because we do not have human eggs exposed to chemotherapy, especially with these alkylating agents. So we plan a final experiment to test the in vitro NMN supplementation for immature human oocytes at advanced maternal age, because we do not have the chemotherapy damaged one. So we will happy, at least, to assess the effects on aging. For doing that experiment, we followed the GB rescue approach. That means that in a standard stimulation cycle, some of the oocytes still remain at the GB stage, and some of them, if we let them 24 hours, they are able to reach the metaphase 2 stage. So what we did for these experiments is we enrolled 54 younger patients, mean age 27 years, and 27 advanced maternal age women with a mean age of 41 years. We collect the GBs from these patients that do not contribute to their IVF cycle, and we randomize in a 1-to-1 ratio for each patient those GBs to the standard GB rescue culture media or to the same media supplemented with 100 micromolar NMN. And what we found in this experiment is first that the NMN supplementation rescues immature human oocytes from advanced maternal age patients by improving nuclear competence from 25% to 50%. This means that we are increasing the probability of these damaged eggs to reach the metaphase 2 stage. But for research, we cannot create embryos, but we want to check if they are able to be fertilized. So we use the parthenogenetic activation approach, and as you can see here, NMN treatment was able to increase the normal parthenogenetic activation in these already aged eggs from 14% to 46.2%. And interestingly, as we observe in the animal models, seems that the NMN supplementation also has some deleterious effects on the ability of the young oocytes exposed to NMN to get this normal oocyte activation. In conclusion, from this experience, NMN supplementation during a whole folliculogenesis cycle increases NAD levels, reduces reactive oxygen species, and modifies mitochondrial distribution, leading to improved spindle assembly, fertilization rates, embryo development in chemotherapy-damaged oocytes. When NMN is restricted to the later stages of folliculogenesis, it could also increase the developmental competence of oocytes, although the effects were not as potent as when we provide the supplementation during the whole folliculogenesis cycle. The in vitro NMN supplementation rescues immature human oocytes from advanced maternal age patients, as well as boosts nuclear competence and parthenogenetic activation. And one last conclusion, which for me is quite important, which is that in a healthy, balanced system, like the young, healthy animals or the young GBs recover from healthy patients, this additional supplementation might produce deleterious effects. because on aging or chemotherapy, we are repairing oxidative damage. But when the system is balanced, this additional energy could lead to reductive stress. As a take-home message, the demand of techniques to also improve oocyte quality is increasing both in the healthy and diseased ovaries due to postponed maternal age when the quality of oocytes decreases. We have seen some pathways to improve oocyte quality, but there are so many other options that should be explored, including, for example, the use of senolytic drugs, the modification of some DNA repair-related molecules to allow the eggs for being repaired, or the pharmacological drugs to treat telomere shortening. I would to thank you all for your attention, and to my team in IBI Foundation, IBI Valencia, and Hospital La Fe, and especially to my mentor, Professor Pellicer. Thank you.
Thank you, Sonia. And we can move on to the next speaker.
Okay, my pleasure is to call to the podium Giovanni Coticchio, of course, one of the fathers of this wonderful meeting, so I think we have to thank you twice. Thank you.
Thanks a lot. Thank you for introducing me, and thank you for staying here and listening to me until relatively late. So I think if some of you have noticed this title before the meeting, you may ask yourself, what this guy is going to talk about, the oocyte cortex. I mean, we knew about the brain cortex, the ovarian cortex, but the oocyte cortex is something really alien to many of us. And in fact, I'm trying to convince you that the cortex, in fact, represents a major functional domain in the oocyte. And not only myself, but also other colleagues have elaborated some concept that I'm going to share with you today. So, the outline is going to be very straightforward. I'm going to talk a little about structure and function, and I'll describe a major player in this stage of the cortex, which is the subcortical maternal complex, and then I will somehow highlight some possible practical implications of this functional domain for human IVF. So, structure and function. What is actually the cortex? It's present more or less in all cells. It provides mechanical stability and defines cell shape. It is located just beneath the cell membrane. In the oocytes, the cortex is particularly rich, especially in actin, and you can see stained in red in this GV oocyte. However, when you expand the molecular structure of the cortex, you may appreciate how it is composed of different elements, not only actin, but also myosin 2, with which actin interacts and regulates tension and contractility of the cortex itself. There are also a plethora of functional molecules, such as a few here are mentioned, ARP2 and 3, which is a molecule that somehow nucleates and regulates assembly and disassembly of strings of actin, and there are also cross-linkers. So altogether, these components form a thick layer of up to a micron in the mammalian oocytes that can regulate cortical tension, and that is important for the oocyte function per se, and possibly also for the embryo. So let's look at other concepts which are key to understand the cortex. So initially, once was being started to be studied, the cortex was considered something pretty, yes, robust, but rather inert. But in fact, as the studies has progressed, a new concept has been developed. The cortex is not a static structure. It is very versatile, very dynamic, and it should be seen as a kind of a protection for the oocyte, or simply a modality by which the oocyte, the largest cell in the human body, acquire a spherical function, but it is entirely functional. So therefore, more similar to this kind of armor-assisted devices that are being employed in human labor. And in fact, I'm going to show you some elements which demonstrate how the cortex is involved in all the major processes gravitating around the oocyte. Oogenesis as a start, cell shape, which is important for the oocyte, cell design, how the cell is organized inside, meiosis, fertilization, early embryo development, everything involving the life of the mammalian oocyte. And the cortex is important almost from the beginning of the human, the mammalian oocyte development. Here you see a preantral follicle, and you can appreciate how, looking at the confocal microscopy picture at the right-hand side of the slide, how the oocyte is somehow contacted by a projection from the surrounding follicle cells. And the cortex in this interface between somatic and oocyte interface is crucial. And if we go a little bit deeper in this description, we see here in this cartoon the graphical representation of the follicle, and then how the surrounding somatic cells, the cumulus cells, projects almost axon-like projection of the cells through the zona pellucida and making contact with the cell surface, the oocyte cell surface. And that takes the form of a very specialized interaction between these transzonal projections, so-called, and the foot of those projections to the oocyte surface. So there is a very specialized organization between the terminals of these projections and the oocyte surface. And the cortex play a major role in that. You can see another micrograph, and you can really appreciate the numerosity of those projections coming from the somatic cells to and moving and projecting towards the surface of the oocyte. And the importance of the cortex is due to the fact that all these interactions that takes the form of gap junctions, of release of small particles and other modalities by which somatic cells and oocyte cross-talk with each other are controlled by the cortex. The cortex is somehow the controller of those interactions modality between somatic cells and the oocyte. And I was saying before that the cortex is very dynamic, and in the first place I would like to show you with this micrograph how the cortex is more or less homogeneously organized in the GV oocyte, the upper side of the micrograph part, and then as soon as a polar body is emitted, or as soon as, I should say, the chromosomes are moved towards the outer part of the oocyte, the cortex acquires a polarized organization. Now, polarity is important, an accumulation, in fact, of actin and other molecules near the part where the chromosomes are located, because that is functional to the modality by which, in this case in the mouse, a penetration of sperm is prevented in the surroundings of the maternal chromosomes, because if that happens, there is a high chance for the oocyte to undergo monoprolucular fertilization through a combination of the two maternal chromosome components in a single pronucleus. So by doing this, by having polarization of the cortex and a specialization of the oocyte surface, which prevents penetration in that part of the oocyte near the chromosomes of the mother, the monopronuclear fertilization is prevented. It is important also to know that these changes in the oocyte surface, which take the form of macrovilli near the chromosome part of the oocyte surface, changes with the aging of the oocyte in vitro. So the more we keep our oocyte in vitro, the more pronounced are the changes affecting the cortex. And this is something that we should keep in mind. As far as the human oocyte is concerned, this polarization or actin distribution is limited. It's still there, as you can you notice in this kind of densitometry picture. However, is less pronounced compared to what we see in the mouse oocyte. And that has implication because a low marginal polarity, if you like, of the human oocyte does not prevent the penetration of sperm in a contact of sperm with the oocyte in the surroundings of the maternal chromosomes. And we have morphokinetic evidence of that. By time-lapse microscopy, we can, in very specific conditions, experimental conditions, to follow in standard IVF the penetration of the sperm through the zona, and in some cases contact between the sperm and the oocyte in the surroundings of the polar body where presumably maternal chromosomes are located. In that case, if that happens, there is a high chance for the oocyte to undergo monopronuclear fertilization. We are learning more and more how those zygotes may be normal, but many of those zygotes have been discarded and eliminated so far. So there are implications for human, the practice of human art. And that dynamism is not limited to distribution of the cortex. Also, throughout meiosis, we have a reduction in cortical tension, and that is functional to what it follows later. You can also appreciate how near the position where the chromosomes are located, there is this sort of cap, a kind of accumulation actin surrounded by myosin in different colors, red and green respectively. And that is the cellular manifestation of the polarity of the oocyte. Now, this accumulation of contractile molecules, in fact, can also create a different cortical tension in different domain of the oocyte, as you can appreciate in this cartoon. I mean, the higher the concentration of molecules, the higher the pressure exerted in the surrounding cytoplasm. And when these interplays of polarization, accumulation of molecules, cortical tension, and pressure on the oocyte cytoplasm is somehow affected, there are implications. For example, let's put the case of a perturbation by which, instead of producing a relatively small polar body, a much larger polar body is produced. And this is seen occasionally in human oocyte. And in those cases, actually, I've just put some dotted lines to help you looking at the oocyte and the relative size of the polar body. You can appreciate how the spindle organization is completely disrupted. So that is one of the consequences of dysfunction of the cortical tension interplay during polar body emission. Another possible situation is when there is a reduced polar body second tension during normal fertilization. I'm going to show you an oocyte that has been injected. You can say that this is a second polar body, fine, apparently, but then the polar body is reabsorbed by the oocyte and you have a pre-pronuclear fertilization. So when the polar body does not maintain sufficient cortical tension, it is taken up again by the oocyte. And these are things that affect our, the efficiency of our effort in our laboratories. And I would like also to show you how there is an interaction between the chromosomes and the cortex itself. Now, we think that chromosomes have two major functions. One is, transmission of genetic information. Second, its expression. It's not just that. Because if you really look at the mechanisms of cortical formation, cortical regulation, now we know, I'm cutting a long story short, we know that chromosomes are able to emit a biochemical message, a gradient based on GTPases, RAN GTP and CDC2 GTPases, that ultimately are able to organize a polarized actin cap, and also to, at least in the mouse, generate a microvilli free zone. And that is another less known function of chromosomes. So not just genetic materials, but also somehow kind of architects of the cell organization, which is interesting per se, I believe. And there is also some preliminary experimental evidence that when, again, experimentally, the cortical tension of oocyte is somehow altered by using specific constructs. For example, these molecules are regulators of the cortical tension interplay. And when those molecules, those proteins are illegitimately, if you like, expressed in mouse oocyte, that interfere in the interaction between actin and myosin. And then the final effect is a kind of perturbance of how chromosomes are captured by microtubules in the formation of the meiotic spindle. So when compared to the controls, when you introduce these altered molecules which regulate actin and myosin interactions, there is a kind of perturbance of the modality by which chromosomes are captured, with inevitable effect in terms of generation of neoploidy. Another major player I was mentioning before at the beginning of the talk, the so-called subcortical maternal complex. So these words define its constitution. It's somehow near the cortex, attached to the cortex. It is maternal, so laid over the entire oogenesis, and it is a complex. In other words, formed from a number of proteins. The proteins are diverse. Some of them, the most important, are listed here. First identified in the mouse, as always, but then confirmed in a number of mammalian species, including the human. So human oocytes have this complex in place. And the complex is now known to regulate a number of very fundamental processes or properties listed here: zygote gene activation, symmetry and asymmetry in the cell design regulation, spindle assembly, etc., etc., etc. And it is also important to note that it doesn't remain in place only in the oocyte life. It is transmitted to the embryo, and from this micrograph you can appreciate in the mouse, but the same applies to the human, that these subcortical maternal complex elements, flopped matter, etc., etc., which are representative of what happens to the entire complex, are transmitted throughout the entire process of blastocyst formation. And when those molecules are somehow affected by mutation in their genes, and therefore abnormal proteins are produced, the effects are dramatic. They are listed here, but in the vast majority of cases, they correspond to embryo arrest at some stage from zygote formation to blastocyst formation. And this is not just basic research. This has been shown to be happening in human development and causing embryo rest. For their very nature, those mutations affecting cortex elements are not very common. Otherwise, human reproduction simply would not be possible. But they exist, and they explain rare or relatively rare cases of embryo rest in our patients. So, an important aspect of exploration in the genetics of human development. And if we still focus on the embryo, there are other aspects that may be relevant to the cortex of not in this case not of the oocyte but of the blastomeres. Now we've learned before how chromosomes may affect the cortex and somehow modulate how the cortex get organized in order to extrude a polar body, either first or second polar body. Now, there are situations experimentally tested, but possibly occurring during normal development, by which chromosomes, for some reason, during cell divisions, may somehow lag behind, may be somehow late in their process of cell divisions, and may be lingering and staying for too long and too close to the blastomeres, forming blastomeres. So these chromosomes here, this chromatin is too close to the cortex and remains so for too long. If that happens, there is a kind of illegitimate activation of the process that brings the formation of a polar body. And that, if happening, may justify fragmentation in early cleaving embryos. And this is exactly what happens in a mouse model. So it is possible that human fragmentation of human embryos may occur because of a similar process in which chromosomes stay for too long, too close to the surface of cells. This has been shown by Cristina already, so I would not insist on that, just stressing the fact that the physical forces, literally speaking, shape the organization of the embryo, even at the transition between the morula stage and the blastocyst stage. Practical implication for human IVF. We know that the cortex is affected in some morphologically abnormal oocyte. That may be a possible reason why those oocytes perform better than normal morphology oocyte. And we know also that with vitrification we disrupt the connection between somatic cells and the oocyte. So if we want to, for example, assess cryopreserve GEVs, probably it's a better idea first mature and then cryopreserve. And finally, there are modalities by which we can measure mathematically the distortion of the oocyte surface and the zona pellucida. In fact, not only is the cortex or the oocyte, and we can measure it, the distortion as an effect of negative aspiration, and make a prediction of oocyte quality. And this kind of approach can perform better than experienced embryology in predicting embryo development. And finally, I would like to recap what we said so far and coming to an end to this talk. So, cortex is a kind of micro machine that is not just necessary to give shape to the spherical organization of the oocyte. It is an entirely functional and dynamic structure. It is key for fundamental process of the oocyte, maturation, fertilization, and the following embryo development. There is a potential for as a prognostic tool in assisted reproduction, if we find a way by which we can measure, the tension of the cortex and try to follow up how this parameter is associated to embryo development, it could be a relatively non-invasive modality by which we can assess embryo quality. And there are further future perspectives in that respect. And if you would like to find out more about this topic, there's going to be this paper published in Human Production. Thank you.
Okay, so let's thank all our speakers, and I'll ask the speakers if they can come to the podium, and we can open the discussion. And I think we have at least the possibility to for a question for each of the speakers. I've got many, so but I'd let you first. No, you go ahead, Laura.
I have to choose. I have many questions. One
Me too,
one,
yeah.
I have to choose which one. So maybe Pasqualino, because you're
Okay.
the first one. It's so exciting to see that from veterinary research we can find really tool for us for human IVF and life. I mean, the possibility to cryopreserve gametes in a different way that would be much more easier to handle, to transport. It's something really disruptive for us. And how far are we from that for human sperm? And what are the main obstacles to obtain the utilization of sperms?
Lino, could you please come to the podium? Yes, thanks.
Thank you, Laura, for the kind words. Yeah. Well, as far as the large animals are concerned, I think, we are ready to use, dry spermatozoa normally. I would bet if you take dry human spermatozoa, they will make pronuclei and embryonic development as similar to a fresh one. I really would like to see this because, human spermatozoa are smaller than ram ones. They have less DNA because you got 46 chromosomes, a sheep one, the 54. Well, I would like, actually, we are doing all these follow-up experiments just to prove that, storing a gamete in a dry form doesn't affect, epigenetic or genetically the gamete. I think the message you have so far obtained from our animals, you have now something like 15. The flock is growing. I think they are quite optimistic, Not so sure. I know that in Japan, because I asked these guys to inquire in all the Japanese IVF, whether they're interested or not in dry storage. But they understand that, cryo works so fine. What is the reason to change it?
Because the cryo room is very expensive.
Yeah.
And it's very difficult to manage. And the transportation of sperm is very expensive also, and will make a lot of transport. So, dry sperm would be much easier to handle.
So let's make the first dry baby on the world.
But Lino, I think I missed it. But what is the success rate of using dry sperm compared to fresh sperm?
In the sheep?
In the sheep, yeah, of course.
Yeah. Fresh sperm and dry sperm euphorics is the same.
Is the same.
Absolutely. Okay. So we have 13% blastocyst rate from fresh spermatozoa. 13, 1, 3. Yeah, 13, ICSI doesn't work very well also only on horses, Cesare Galli, all these guys. Yes. But if you apply it on cattle, it's the same. Pigs, goats, they all perform in the same poor way, I think. Yeah, I think we are ready. Going back to the protamine, yes, I feel very good because, as a cloner, I really wanted, that was my dream. I realized the oocytes cannot handle the chromatin structure of a somatic cell. The only way was to trick him, the oocytes, providing the somatic cell the organization that you have in the spermatozoa. That's the only format of DNA the oocyte is able to read. So I was very proud of it, but on the other side, I'm quite frustrated because, I didn't prove it till the end. The Chinese did, unfortunately, but, it happens many times. Thank you again.
Any more questions? Yes, please, go ahead.
Thank you, everyone. Very brilliant and good presentation, but I have a question for Sonia. So I would like to know if you think it's more reliable to use the NMN as an oral administration for patient directly, or you think the best way to follow is this in the culture media?
Well, thanks for the question. I think that both. I mean, it depends on the situation and the prognosis of our patients. They are not excluded. We can just try to improve how the oocytes develop, but even though for some cases with good ovarian reserves or non-expected bad oocyte quality, maybe we can use them on the in vitro culture medias, because they are working through a different mechanism, and hopefully we will have soon some data on the oral supplementation in patients, not only in mouse. But I mean, they work by different ways, so they should not be excluded. We can maybe use them both. Thanks.
Any more questions? I've got one for Maria José. Excuse my very huge ignorance about mitochondria, but I always thought that they are so big. And I was wondering whether mitochondria are exchanged between cells, whether, for example, cumulus cells can transfer, maybe through transzonal projections, a mitochondria to the oocyte or vice versa, or are there other mechanisms of mitochondria transfer from cell to cell?
Yeah, there are actually. We've seen that when we were doing this review of mitochondrial transfer, we observed that this mitochondrial fusion between, the transport between cells is something that is real, happen in many tissues in order to recover some damage in a certain tissue. So I think this transportation between cells is something that happens. I'm not aware of this transportation between cumulus cells and oocyte. It might be, yes, but I really don't know.
Does the size of the transzonal projection allow the...
Good question. I don't know for sure. It's true that maybe if this may happen, the mitochondria may adapt the shape to go through the transzonal junctions, but honestly, I don't know. But I do know that this transportation between tissues in the same tissue between cells is something that the organism use for repairing mechanism between cells. So this happens in the organism.
Thank you.
Yeah.
Thanks a lot.
Thank you.
Cheers. Any more questions? I've got one for Giovanni. Yes. Yeah. Giovanni, is there a correlation between the size of the cortical cap and the size in diameter of the PB1? And in turn, could there be a correlation between the cortical cap size and the oocyte developmental competence?
Yes, we know something about this point in relation to the mouse species.
Yeah.
So in the mouse, if you mature in vitro a GV oocytes and release it, remove the cumulus cells, the GV somehow is traveling from the cortex to the central part of the oocyte. It undergoes breakdown, and the chromosomes are transferred back to the cortex. In this atypical situation, a larger cortex accumulation of proteins is generated and a larger PB is emitted. Unlike the normal situation in vivo, where presumably the GV is always located cortically and there's not this back and forth of chromosomes between the center of the oocyte and the cortex. In the latter case, in vivo, the polar body is smaller. We don't know exactly what happens in the human, but it's very likely that if you have a large polar body, occasionally, it's not common to see that in the human. This is due to maybe incorrect distribution of chromosomes which may be occupying a larger space portion, and that may affect a larger part of the cortex nearby. I mean, this is my assumption. There's no proof of that.
Okay.
And the other thing was, no, I can't remember.
Whether there is a correlation with the oocyte developmental competence. We can
correlate
these sizes.
I mean, in the mouse, yes, because especially in the mouse, in which the oocyte is smaller, you have a larger removal of cytoplasm through the emission of large polar body. So what is left for the oocyte is a smaller quantity available for the building of the embryo. Okay, that is the possible explanation. In the human, oocyte with polar bodies very large are very rare, but when occurring, there is some inference that they may have a lower developmental competence. That is what we know at the moment. Okay.
I think that time is over. Are you okay? No more questions?
Coffee break.
Okay, coffee break. See you in a while.
Thanks.
Thank you, everybody, for a wonderful session. Thank you so much.